This is the flipped lecture over mutations. After I finish instructions, please pause the video and set up Cornell notes on pages 8 and 9 in your journals. You've also been given a fill-in-the-blank companion to go along with the lecture. Be sure to write your guiding question at the top of page 8, which is, explain when mutations are beneficial for an organism. You should be able to answer that question in your notes summary at the end of the lecture. Please complete the lecture before you come to class on Tuesday, as well as we will be doing a mini lab called Mutation Telephone. As with all flipped lectures, please feel free to pause, rewind, take breaks, or break up the lecture to fit your study needs. If you are struggling, see Coach Merrifield or Miss Priest for tutorials before or after school. Go ahead and pause the video and set up your journal, and resume when you are ready. Let's make a change. Your learning objectives for this lesson are: students should be able to list the different categories or types of mutations, describe some possible effects of mutations, and explain the role of mutations in genetic syndromes. Mutations can be classified several different ways. In this lesson, we'll focus on sorting mutations by their effects on the structure of DNA or a chromosome. So for this categorization, they can be categorized into 3 main groups, each with multiple specific types. The 3 general categories will be discussed here shortly. So what are mutations? The effects of mutations may range from nothing to the unviability of a cell, meaning it's not viable or it's basically cell death. All mutations affect the proteins that are created during protein synthesis, but not all mutations have a significant impact. If you take a look at the photos here, those are all examples of mutations in various species. So again, what are mutations? Take 30 seconds, pause the video if you need to, and write down what you think your definition of a mutation is. All right, let's continue. So, how did Cyclops from the X-Men get his superpowers? How did Hulk and Spider-Man get theirs? Well, Cyclops was born with the mutation, and the Hulk was exposed to gamma radiation, and Spider-Man was bitten by a radioactive spider. So again, I ask you, what is a mutation? Mutations are changes in the DNA of a cell. Some have a positive effect, a negative effect, and some have no effect at all. Today we will be discussing some of the science behind how these mutations could happen. While the superpowers are abilities may be fictional, it is true that mutations can have significant impacts on people, and evidence exists that radiation exposure can lead to an increased rate of mutations. Let's discuss the different types of mutations, where or how they can occur, and we will also look at some environmental factors that can influence the rate of mutations, and finish by looking at some possible effects of mutations. What are the causes of mutations? Well, mutations naturally occur over time, which is the underlying cause of evolution. As we can see, evolution is a very slow process with a net benefit to an organism. But some environmental factors may influence or induce additional mutations. These induced mutations often lead to harmful diseases such as cancer. So causes might be mistakes in replication in the S phase, mistakes in protein synthesis, or external agents we call mutagens. Exposure to certain chemicals is one environmental factor that may induce DNA mutations. Typically, anything that we identify as a carcinogenic may cause cancer. It has negative side effects on DNA and may lead to cancer. This includes the chemicals found in cigarette smoke as well as those found in meats cooked on the grill. These chemicals belong to a larger class called mutagens, meaning they can lead to changes in genetic material. Chemicals are not the only types of mutagens that we encounter. Physical mutagens also exist in the environment, namely radiation. Ultraviolet radiation from the sun can damage genetic material by changing the properties of nucleotides in the DNA. So overexposure to the ultraviolet radiation is known to lead to skin cancer. X-rays and gamma radiation are also physical mutagens and forms of ionizing radiation. This means that these types of radiation possess sources of energy to remove electrons from atoms, thus forming ions and affecting how different biomolecules interact. While a typical dose of X-rays received during a medical procedure is low, it does marginally increase a person's cancer risk. Alternatively, retroviruses such as HIV naturally experience mutations at a much higher rate than other organisms. This can be attributed to the fact that they possess RNA instead of DNA. The process by which RNA is copied and replicated is not as precise as that of DNA. Therefore, by the time our immune system is adjusted to fight a virus like HIV, the HIV virus is already mutated again, and the immune system must start over. The mutations in the HIV's RNA lead to alterations in the protein markers on the virus that the immune system targets. And if the target is always changing, it's almost impossible for the immune system to remove the virus. Only mutations in sex cells or your gametes will be passed on to your offspring, though. So, what are the three categories of mutations? You have small-scale mutations, which would be your point mutation and your frame shift mutations. Then you have what we call large-scale mutations, which would be your chromosomal mutations. So your small-scale mutations are those that affect the DNA at the molecular level by changing the normal sequence of nucleotide base pairs. These type of mutations may occur during the process of DNA replication during either meiosis or mitosis. Three possible types of small-scale mutations may occur. We'll get into those here in a minute. Large-scale mutations are those which affect entire portions of the chromosome. Some large-scale mutations affect only single chromosomes, and others occur across non-homologous pairs. But some large-scale mutations in the chromosome are analogous to the small-scale mutations in DNA. The difference is that for the large-scale mutations, entire genes or sets of genes are altered rather than only a single nucleotide of the DNA in small scale. So let's get into small-scale mutations. For example, point mutations or substitutions. Point mutations is a change in a single point or a single base on the DNA sequence. So substitutions are much more subtle than frame shift and have three possible effects. Again, a substitution is one base is changed to another. It affects no more than a single amino acid. So the three possible effects here are silent. The nucleotide is replaced, but the codon still produces the same amino acid. Missense, the codon now results in a different amino acid, which may or may not significantly alter the protein's function. Nonsense, the codon now results in a stop command, truncating the protein at the location where the mutated codon is read. This almost always leads to the loss of protein functionality. These mutations may occur anywhere in the DNA, so the effect of the mutation really depends on its location. If the mutation occurs in a gene, the result is an altered protein. But if the mutation can occur in a non-genic region of the DNA, then the mutation has no effect on the organism. So this is substitutions, also referred to as mutation, point mutation. Substitutions occur when a nucleotide is replaced with a different nucleotide in the DNA sequence. The most common substitutions involve the switching of adenine and guanine, or cytosine and thymine. Since the total number of nucleotides is conserved, this type of mutation only affects the codon for a single amino acid. One base replaces another, and it changes one amino acid. Frame shift, our second small-scale mutation, equals when the single base is either added or inserted or lost, meaning deleted. There's a shift to the reading frame of the genetic message. So frame shift mutations, which are insertions and deletions on genes, have similar effects. When a nucleotide is added or removed from the DNA sequence, the sequence is shifted and every codon after the mutation is changed. That's why they are the most harmful mutation. This results in severe alterations to proteins that are encoded by the DNA, which can lead to a loss of functionality for those proteins. So here's an example of an insertion. In the addition of a nucleotide to the DNA sequence, similar to a deletion, insertions are also considered frame shift mutations and alter every codon that is read after the mutation. Deletion. A deletion is the removal of a nucleotide from the DNA sequence. Deletions are referred to, again, as frame shift mutations because the removal of even a single nucleotide from a gene subsequently alters every codon after the mutation. It is said that the reading frame is shifted. Loss of part or all of the chromosomes. So the fat cat ate the rat. Delete or remove the H. It changes the entire amino acid. Now I have tef, act, ata, tet, her, at. Does that even make sense? Every codon after the mutation is changed. The change in the number of nucleotide changes, which ones should normally be read together. Large-scale mutations, our third type, chromosomal mutations, involve changes in the whole chromosome. It occurs during the division of sex cells or in meiosis. It affects entire portions of the chromosome. Some large-scale mutations affect only single chromosomes, others occur across non-homologous pairs. Entire genes or sets of genes are altered rather than only single nucleotides of the DNA. Mutations involving multiple chromosomes are likely to occur in meiosis during prophase 1. So what are the 4 types of chromosomal mutations? Chromosomal mutations are large-scale mutations, and the effects of large-scale mutations are more obvious than those of small-scale. Duplication of multiple genes causes those genes to be overexpressed, while deletions result in missing or incomplete genes. Mutations that change the order of the genes on the chromosomes, such as deletions, inversions, insertions, and translocations, result in genes that are close together. Astrocytoma, which is a type of brain tumor, is the result of a deletion that creates a new fusion gene that permits the cells to become cancerous. Often a large-scale mutation leads to cells that are not viable, meaning the cell dies due to the mutation. This is especially true with non-disjunction mutations in gametes in which entire chromosomes are missing or extra. In humans, when the gamete from a male merges its chromosomes with the gamete from a female, the offspring receives 23 chromosomes from each parent to form 23 homologous pairs. However, when one of the gametes has a non-disjunction mutation, the resulting offspring end up with only one homolog. When we talk about the chromosomes, that X, they only end up with one part of the X. I'll show you what I'm talking about in class. So they only end up with one homolog in a pair, or with 3 homologs in a pair. So if they have 1, it's called monosomy. If they have 3, it's called trisomy, because mono is 1, tri 3. Most of the time, these offspring are not even viable. The ones that do result in viable offspring will possess some noticeable differences due to the extra or missing chromosomes. This alteration leads to a permanent syndrome in the offspring. The most well-known syndrome is trisomy 21, an extra 21st chromosome. This particular non-disjunction mutation leads to Down syndrome. Several other syndromes are caused by these mutations. If you're interested, I would suggest research some syndrome caused by an extra or missing chromosome and just write a short paragraph detailing which chromosome is altered and what effects that mutation causes, if you're still struggling with that. All right, so let me go back actually one. So the 4 types of chromosomal mutations are deletions, where part of a chromosome is removed or left out; duplication, DNA sequence or the base is repeated; inversion, part of the chromosome breaks and rejoins in reverse, so it basically flips upside down; translocation, portion of one or more chromosomes breaks off and rejoins an unrelated chromosome. So let's take a look at these in picture form. So again, a deletion is a single chromosome mutation, and it's the loss of one or more genes. If you look here, here's your original chromosome, and in the new chromosome after their mutated chromosome, B, gene B has been removed or deleted. If you look here, duplication, gene B has now been duplicated. It's a single chromosome mutation and the addition of one or more genes that are already present in the chromosome. Inversion is where the original chromosome, the mutated chromosome, inverts, or it's the complete reversal of one or more genes. Sorry. Oh. Go back here to inversion. Sorry. It's the complete reversal of one or more genes within a chromosome. The genes are present, but the order is backwards from the parent chromosome. So here's my parent chromosome, A, B, C, D, E, F. Over here I have A, E, D, CBF. That's my inversion. CB has flipped and reversed itself with ED. Translocation, multiple non-homologous chromosome mutation. Chromosomes swap one or more genes with another chromosome. So these chromosomes are swapping, or these genes are swapping with these right here. So I have ABC, DEF, GHI, JKL. ABC, JKL, these are non-homologous chromosomes, okay? And now I have JKL, and then I have GHIDEF. So they have translocated or swapped from one chromosome to another. One that I didn't talk about right here was insertion. We talked about deletion where we take it away. Insertion is a multiple chromosome mutation. One or more genes are removed from one chromosome and inserted into another non-homologous chromosome. So here's the parent or normal chromosome, and here L, the gene L, has been inserted to that parent chromosome. It can occur by an error during the prophase 1 of meiosis when the chromosomes are swapping genes in crossing over, a process called crossing over to increase diversity. All right. What are the types of point mutations? Well, here are your types of point mutations: substitution, insertion, deletion, and inversion. What we did here was we used examples of letters. Here I have beast. Well, if I substitute F for B, I have feast. Then if I insert an R, I have breast. If I remove A, I have best. If I invert the S and the T, I have beats. Pause the video and examine the different types of mutations in the words. What a difference a letter can make, huh? All right, what is the significance of mutations? Well, mutations can be harmful. They can benefit. So some harmful mutations, they can cause harm to organisms, making it less likely to survive. They can cause genetic disorders, and they're also associated with cancers. Now, benefits. Benefits, DNA changes that help an organism survive better. For example, polyploidy, meaning organisms with extra sets of chromosomes, which is good in plants. It gives them extra leaves, so more sunlight, extra flowers, things like that. So while mutations occur naturally over time, biological engineers are able to genetically modify various organisms. Humans have been genetically modifying plants and animals for thousands of years. We've actually accomplished this by selectively breeding or interbreeding in order to produce and improve specific traits, such as breeding watermelons to be larger and have fewer seeds, or breeding chickens to have more white meat and more breast meat. With the advancement of technology, engineers can directly manipulate the genetic code of a plant and animal. Some examples of genetically modified and controversial organisms include disease-resistant papaya, vitamin A-rich rice, and drought-tolerant corn. But we'll dive into that in our next unit, genetics. So, silent mutations change nothing. Mutations are the source of genetic variability and can be highly beneficial. Use this picture to answer your guiding questions and from the beginning of the lecture, and do not forget to complete your notes summary. Make it 2 to 3 complete sentences. Show's over. See you in class. Again, if you have questions, see Coach Merrifield or Miss Priest before or after school for tutoring.